Hello, class. So this is the biosynthesis of amino acid series. So this is a three-part series, and this is the first session of the series. And what we will be talking about today is nitrogen fixation, microorganism use ATP and a powerful reductant to reduce atmospheric nitrogen to ammonia. So the nitrogen in amino acids, purines, pyrimidines, and other molecules ultimately come from atmospheric nitrogen. And you have this nitrogen that ends up getting reduced down to ammonia. And so higher organisms are unable to fix nitrogen. So this conversion is carried out by bacteria. And so what happens in order to have this biosynthetic process, it starts with the reduction of nitrogen again to that ammonia, and this can actually be done in an industrial process, which was found in 1910, where you have a fixation of nitrogen, and it's typically carried out by mixing hydrogen gas over an iron catalyst at 500 degrees Celsius and pressure of 300 atmospheres. So that is covered right here. So you want to take note of that. But just know in nature, we have other organisms such as bacteria that can do this nitrogen fixation as well. And so in order to have this nitrogen fixation, is catalyzed by a nitrogenase complex. So it's made up of two proteins. So this protein is the iron protein or reductase. This is where all of the electrons are stored. And so from there, you have this ATP hydrolysis. So you see that a phosphoryl group is released. So this is an ATP dependent type of reaction. And so then the electrons go over to this protein, which is the molybdenum iron protein, and also considered nitrogenase. And that is what actually reduces the nitrogen to ammonia. And this reaction, which is written down here, requires 8 electrons in order to happen. And so, also looking at this reaction that's listed at the bottom, also note that with 8 electrons, remember this is an ATP hydrolysis type of reaction to carry over that electron over to the other protein. Um, in order to do that, how much ATP does you need? And so 2 molecules of ATP are hydrolyzed for each electron that is transferred. Therefore, since there's 8 electrons that is being transferred over in order to have this reduction of nitrogen to ammonia, that means you do 8 times 2. And there is a requirement of 16 ATP in order for this to happen. And so oxygen is actually required for the oxidative phosphorylation to generate the ATP that's necessary for nitrogen fixation. However, this nitrogenase complex is actually sensitive to inactivation by oxygen. So it can't be around oxygen, but it needs oxygen for this generation of ATP. So what happens is ATP synthesis and nitrogenase function simultaneously in certain plants called leg, leguminous plants, and it's maintained a very low concentration of free oxygen in the root nodules. So the location of nitrogenase can be there to actually have this nitrogen fixation that occurs without worrying of too much oxygen is actually inactivating it. And so this looking at this reductase protein, so that's the one that's yellow to the left, also called iron protein. What it has, you can see in the structure, the little blue balls, not blue, but green ball is iron, yellow is sulfur, and then we have a representation of amino acid residue, which is cysteine. And so you have this cluster, this four iron, four sulfur cluster that is within the complex of the reductase enzyme. And so both reductase and nitrogenase components of the complex are iron sulfur proteins. So they both have iron and sulfur, which is once again this reductase and nitrogenase. They both have iron in there, sulfur. And so the role, once again, for this reductase P cluster of four iron and four sulfur is to store electrons. And remember, this is storing electrons for it to be transferred over to the nitrogenase, and this nitrogenase does that reduction. So I want to just keep repeating that just to make sure things are locking in. And so from there, you have this whole nitrogenase cluster. And so remember, for the reductase, it has this iron center, which is a P cluster. And remember, you have these iron and sulfide, and then you have these cysteine residue. So each P cluster contains 8 iron atoms and 7 sulfides linked to the protein by 6 cysteine residues. And so as far as the molybdenum iron, so that contains one molybdenum atom, which is located here. For purple, you have 7 iron atoms, which is green, notated here. And then you have 9 sulfides, and that is yellow, that's located here. And then there is a interstitial carbon atom. This is located right here. And then you have a homo citrate, which is located here and is linked to the protein by one cystinate residue and one histidine residue. So you see your histidine residue, and they have some more with the cysteine residue. And one other thing is these linkage between the two proteins is by a um, joint 3 disulfide bonds. And so now that we have this ammonia that is generated after the reduction of nitrogen, now we need to take this ammonia and to actually create glutamate or glutamine to be as a source of nitrogen for amino acids, pyridines, and so on and so forth. And so ammonia that's generated by the nitrogenase is a base, and it becomes an ammonium ion in aqueous solution. And so the alpha amino group of most amino acids comes from the alpha group of glutamate by transamination. Glutamate is synthesized from the ammonium ion and alpha ketoglutarate, a citric acid cycle intermediate by action of glutamate dehydrogenase. And so, as far as breaking this down, we have this general term where you have a carbonyl that represents this alpha ketoglutarate. So you have the carbonyls and you have two R groups to the side. That is what this is representing. And then you have amine donor that represents the ammonium ion. And so when the two react together, it forms this shift base. And so from there, it is actually readily protonated, and then you have this protonation that forms, and this is the protonated shift base. And so, like I mentioned before, this is just a representation of alpha ketoglutarate, which is listed right here. You have the ammonium ion, which is that amine donor, and now it's forming this shift base. So this structure is what is represented by this figure that is right here. And then from there, you have uh, you have the protonation that's happening. So that's where the black hydrogen is coming, and then the NADPH that comes in handy because that actually reduces the shift base. And so you'll see that written right here. The protonated shift base is reduced by the transfer of a hydride ion from NADPH to form the glutamate. And so you see that double bond is no longer there, so it is being reduced, and you have that hydride uh, transfer to that carbon. And the protein that's right here, that's in black, that is added to that nitrogen, just like how you see with this representation of the shift base, where you have that hydrogen is added to that nitrogen. It's the same thing here. So you see there was two hydrogens, and now there is three. They just in their figured, I know they did some color coordination, and they kind of left off the black hydrogen to kind of see where it exactly goes, but that is where it is actually located, and that forms your glutamate. And so a second ammonium ion is incorporated into glutamate to form glutamine by the action of glutamine synthetase. And so you can see this listed right here at the bottom, where you have your glutamate. Once again, you have an ATP driven type of reaction where it's a hydrolysis of ATP. So you have a phosphorus group that is being released and actually being bounded onto the the general structure of the glutarate. So that is forming an acyl phosphate intermediate, and then from there you have you have the ammonium ion, or how they have in this form, it's just ammonia, but in aqueous solution, just remember that this is ammonium ion, and so that is attaching and replacing that phosphorus group to now form glutamine. And so our source of nitrogen donor is coming from glutamine and from glutamate. So we just went over how glutamate is made, which is that reaction of ammonium ion with alpha ketoglutarate, and you have to have that NADPH. Remember that the ammonium ion and the alpha ketoglutarate form a shift base, and that shift base is protonated, and also it is the carbonyl is reduced by the NADPH to add another hydrogen transfer to end up making glutamate, which is a source of nitrogen. And once again, as far as when you have glutamate and actually forming glutamine, this is glutamine synthetase. And what happens is you have that ATP hydrolysis, and you have the phosphoryl group um, that is attached to make this intermediate. Then that's where ammonium ion, or ammonia, is used to replace that phosphoryl group. So you see that it leaves off and finally forms glutamine. And I believe that is the end of this lecture.